All right, so I'm going to do my best with this here. There's probably more information here than I will require you to recall, but we will go through all of it, and then through the semester we can discuss which components are the most necessary for you. This is a lecture prepared by Dr. Poston for the 465 class last year. So we see that the motor cortex is really what gives us this voluntary movement, and we are unique in the animal kingdom. We have a more evolved motor cortex, and it really gives us sort of a way to be able to feed ourselves, particularly our arms and hands. So we have a lot of use of our arms and hands that is quite unique among other animals. So primates, certainly, we're not the only ones, but primates, but we have been able to become, if you will, at the top of the food chain largely because of our ability to move our hands or arms, to have this voluntary movement in such a way that is very direct and specific. So, they are. One of the books says that humans can also forage for food by cooking or punching a few buttons on a telephone. And obviously, this is far more advanced than other animals who can't do this for themselves. So although that sounds really simplistic, it's a pretty important part of our survival. All right. So we'll talk about about the primary motor cortex, and this is where the movement is actually generated. So our supplementary motor areas and our premotor cortex, which we haven't really talked about too much, that's really where the movements are staged. Lots of different activities go there, and as the previous lecture mentioned, that's also an area that we see activity when people are considering a movement, but the actual movement itself must occur from the motor cortex and then through certain pathways. There's a lot of terminology that, as we've already seen, is a little bit different from scientist or author to author, and that's just part of the science. It's just something that you just kind of have to bear with and pay attention and understand that, not get overwhelmed by the different terminology. Nobody's expecting you to remember all the different terms for all of these things. If you decide to go into neuroscience, it'll be a useful thing for you to, learn more about, which of course you would if you are involved in the science directly. So we see that when people are talking about the motor cortex, they're often kind of using they may only be talking about M1, and they may only be talking about the primary motor cortex. Sometimes they're also including that supplementary motor area, premotor cortex, etc. So you kind of have to pay attention to the context, just so you have a better understanding of exactly what's being addressed. So we've seen it on the pictures. You've got lots of pictures. It's anterior to the central sulcus, and then of course the premotor areas are further anterior. And so, of course, then again, premotor areas, and this is where some of the mental training and some of that kind of thing can be justified in hypnotherapy or in mental training for sports, is that there is activity in that area of the brain prior to movement. So we see that the primary motor cortex, though, is executing the movement. And they do overlap across the regions. There's lots of different outputs and inputs. So I'm not really going to ask you to go through and understand all of these tracts right now. We're going to go back to ascending and descending tracts in a future lecture, but this will probably be a slide you see again. It's probably not a bad idea to begin to understand that this is not just a direct system. There are a lot of pathways. There's a lot we don't understand, but certainly a lot that we can trace. We see that this on the right, there's some information about the basal ganglia. Dr. Poston's going to talk to you more about. We will talk directly about the cerebellum, of course the thalamus, the relay station there, and so don't get too overwhelmed by that. I think it's kind of a nice diagram that kind of gives you an idea of what's happening. So some of the aspects of voluntary movement are listed below. I don't want to just read these slides to you. I won't lie, I'm in a fairly significant amount of discomfort, so I'm probably going to do this pretty briefly. But there are several differences, and these are differences that I do expect you to know. You should be able to identified that voluntary movements are intentional. It's not something that happens by reflex. It's not a cross-extensor reflex. It's not a stretch reflex. This is something that we've consciously decided to do, and now we have activated the pathways to do so. Context dependent. This is also something unusual. It distinguishes voluntary from involuntary movement. So it depends on the context. If you're looking at the same object, certainly can do different things depending on the context, including no action. If there's a cup with acid and a hole in it, you may not reach out to pick it up. If there's a hot cup, you might pick it up more differently than you would a cup that's tepid or cool. So, it's context dependent. It's not just about the object. And also, we can learn. Our brains are adaptable. They're plastic. We can actually learn and adapt, and we see some of that represented in the areas, in increased gray matter in the cortex. So it's something that we can see in athletes. There are some studies that show that athletes and people that work out have thicker gray matter. And in different areas, certainly, it becomes more robust because the neurons are being used and utilized and stressed, and so they respond by becoming more robust, growing more dendrites, more synapses, so they're more capable of doing the job when called upon. There are a couple of different slides of the homunculus. I would really like for you to kind of look at it. I'm not expecting you to memorize any of that. Just appreciate that there are representation areas. This has been going on for quite some time, first in animals and then during neurosurgery, but now we can use TMS. And so TMS is one of those things Dr. Poston specializes in, and he will tell you a little bit more about it. But we can find there are well-known areas, this is fairly well-known, in the motor cortex that can be stimulated through a transcranial magnetic pulse, and then it directly stimulates a particular muscle that we can then measure the EMG response. So for instance, when I was in the lab, I don't know that I have any pictures to post. I do, but I'm not probably going to show you. Anyway, I was a participant in one of his studies recently, and they were stimulating my motor cortex for my FDI, my first dorsal interosseous, which is a well-known muscle in the index finger that can be measured. So they put an EMG on the belly of that muscle, stimulated that part of my motor cortex with different conditions, and with TDCS, without TDCS, and they could measure the response of the muscle, and that would give them an idea how the pathway was affected by the intervention or the treatment. And so this is something that is very commonly done, and it is in fact how a lot of this mapping has occurred. So a lot of, in the beginning, of course, it was certainly a lot more painful, done through direct electrical stimulation, which is very painful. Magnetic stimulation is a wonderful advent. You'll be learning more about with rock, and it has helped us to learn a lot more about the body without being so inhumane, in fact. But there really is no way to directly, there's a lot of overlap, it's very individualized. In fact, when he was stimulating mine, it was only supposed to be, so of course it's going to be contralateral, right? It's going to be on the other side, because that's the way the motor cortex works. And so they're stimulating my left motor cortex for my right FDI muscle to experience some type of EMG response. And in fact, my left shoulder was also affected, which apparently is very, very unusual. So on my ipsilateral side, no reason. Brock have never seen it before, but it's just, everybody's brain, depending on, could be use, could be injury, could be genetic. So there are a lot of areas of overlap and a lot of differences in between each individual. So that's very interesting, and I might post a couple of slides from Latash because he does go into this pretty nicely. The cerebral cortex is in 6 layers, and they share inputs and outputs, and they're arranged in columns. And so see these pyramidal cells. I call them pyramidal cells. I've heard them pronounced pyramidal, but they kind of look like a pyramid, don't they? So we can see here in this nice picture the 6 layers, the neurons in the cortex. They're basically 2 big groups. We have pyramidal cells and non-pyramidal cells. Non-pyramidal cells are stellate or granule cells. And the pyramidal cells are the ones that we can see there arranged vertically, and they project to the spinal cord. And the granule cells are horizontal, and they are true interneurons. So they're interneurons. They're working to communicate between the different neurons. And so you can kind of think of that in directionality. The pyramidal cells, we can mostly see them in layer 5. And the Bet cells is the most well-known, the most well-studied. Again, it only makes a little bit of the percentage, though, that project directly to the spinal cord. And so there's your picture you can take a look at. All right, so representation areas. Here's an example. So here on the bottom, we can see the figures that have been created from stimulating these areas of the cortex. So we can see that's along the central sulcus. And when that is stimulated, I believe this is probably in an ape, in a great ape, in a primate. A lot of times these are in a primate instead of a human. Anyway, you can kind of see that there are different areas. And I just want you to appreciate, again, that there are different areas. And this, again, is fairly well known, which is something that you as a neuroscientist could study, particularly in this area of motor control. And there's some information about the primary motor cortex and the premotor areas. So we see the different areas that are anterior to the primary motor cortex, or M1, and we also see that they are designated to, well, they are in association areas. So again, this is something that's been studied. We can see that the premotor cortex and the SMA, or the supplementary motor area, they are also kind of divided into smaller functional areas, and they can be, so it's kind of nice, because then you can also tell through diagnosis if somebody is, if a patient population is exhibiting some type of issue in a particular limb or muscle or some area of the brain. Of course, we can trace it forward or backward from this point of the brain, which is very helpful. You know a lot more than you might be surprised. So I'll just let you read through this slide. You can pause this and take some time to read through this slide. All right, so here's some of the confusing terminology. Here's just a list. There are probably others. One of the things I see a lot of are higher and lower motor neurons. That's kind of outdated. So, you can just see that there really isn't a lot of agreement between the different professors and scientists as to what these are supposed to be called. So please don't let that throw you off too badly. Try to maintain, consistency within this course. But it's not wrong. It's just a different way of saying things. So corticomotor neurons, those are coming from the primary motor cortex, and they are forming direct connections. So they are monosynaptic. They're not going through interneurons, and they're going directly to motor neurons, and that occurs in that ventral and lateral part of the spinal cord. So we see the projections from premotor areas. They actually synapse on spinal interneurons. So the M1, primary motor cortex, percentage of those are not on interneurons, whereas the premotor areas, they are synapsing on spinal interneurons, and they're more dorsal and medial. As we have evolved, we have developed more of these corticomotor neurons. Humans have the most direct connections. We have the most direct control. And most of our direct control, a lot of our brain capacity for movement really has to do with our hands and fingers. It's very important. And again, you really have to think of all of the things that we do with our hands and our fingers and our arms that are very specific that other animals can't do, and that might in fact lead to why we have evolved. All right, so we also see the corticomotor neurons. They activate complex muscle patterns, and they diverge. So remember, convergence and divergence. So they are diverging with spinal motor neurons, and those are activating a group or variety of different muscles. So although they are projecting monosynaptically from the M1, They are, in fact, then stimulating multiple muscles. So please take a little bit longer of a look through this information, but again, it gives you the general idea. And for our purposes for the rest of this class, there's plenty to learn without going into just extraordinary detail. So although there's a lot of detail provided for you, I'm not expecting you to memorize all of this. I do want you, however, to be familiar with it and read it. Here's some interesting fun-filled facts for you. If you read that previous slide, which I'm sure you did in a lot of detail, it's actually pretty interesting. They did show that agonist and antagonist muscles were activated. Please note that none of them are activated by the same corticomotor neuron. So separate groups of excitatory corticomotor neurons have to activate their agonist muscle at the same time. So they're separately controlled. Also note that in those pictures, they were not the fine control muscles we were talking about. So hand muscles require more fractionated activity because of all of the things I've mentioned. And those don't have the same patterns of that agonist-antagonist as what we just saw in the previous slide. So here's an example. In the wrist, 42% facilitated more than half of the flexor extensor muscles. This is where it's interesting. In the hand, a single corticomotor neuron cell contracts only 2 to 3 intrinsic hand muscles. So you see why we have to have that entire representation area. We need a lot more fine-tuned control as opposed to controlling hundreds of motor neurons. Now we see that a single cell contracting only 2 or 3. So we see more fractionation of the hand, the digitization of the hand, and of course, again, that's very important for humans and other primates. Well, versus other primates, but certainly primates compared to other species. All right. So we see that wrist muscles are still important, though we still have more fine-tuned control over our wrists than other large muscles like our quadricep group. Also, they're highly measured because they do have some unique, they exhibit some unique EMG patterns. And so we can, sometimes in these sciences, what they do is they put lesions on monkeys' brains to see what they affect. So if you put a lesion on a brain, then you can see what part of the body that part of the brain controls. Now they can actually create virtual lesions that disappear. So as science progresses, they do keep trying to become more humane, and the improvement in technology is helping to save animals in the lab. And certainly there are more studies now also done on humans because of the ease and safety of TMS. So if we see that we section the pyramidal tract, the monkey can't perform these individuated movements. You see that normal, and then after sectioning the pyramidal tract. So we can see that there is some direct control. This is a way that we can show, prove, if you will, that there is direct control from these corticomotor neurons, from the M1 to these muscles, because there isn't anything taking back over. All right, we saw briefly in the other lecture that there are certain populations of neurons in the motor cortex that do have unique functions. So in this case, we do see that some individual pyramidal neurons are used for very specific tasks and movements. And so we see here that we have the wrist being flexed and then extended, and we can see different firing patterns in the corticomotor neurons. So we have different populations working for different movements. Also, there are populations that are about direction. And so take a look at this in a little more detail. We talked about it briefly. They increase or decrease their discharge rate based on direction. And so that might be sort of something you haven't thought of. So when we see a large number of neurons, many cells with a preferred direction can be summed together. They call them population vectors. So some M1 neurons strongly code for movement as well as force. So take a look also at that idea of some neurons code for force, some neurons code for movement. Here's a kind of a famous and often talked about experiment for the population vectors with time. So this monkey was trained to perform this task. He did it over and over again until it was just a natural thing to do. And the population vectors were calculated every 25 milliseconds. And we could see that those population vectors preceded the displacement at a lag of 100 milliseconds. So we could see them fire before the movement took place, and it was very specific. So we see that neuronal population, the discharge rate varies continuously with complex movements, but we also see that they can be expected and plotted. So this is good. So if you know that something is typical or regular in a healthy subject, then you can do similar types of experiments, and if the results are not what is expected, then you can begin to diagnose what area is being affected or has been affected. So this slide talks about an experiment that kind of showed that what was more important, what was the priority, what are the neurons coding for in the motor cortex. So here we see an experiment with a monkey who is moving their wrist in the same direction, in the same displacement, producing the same displacement. And they are measuring these neurons to see what creates activity and what does not. So when there's no load applied to the wrist and the monkey moves, then the neuron fires before and during the flexion. If there's a load applied, then we actually see, there's a load applied that resists the movement, then we see greater neuronal activity, which makes sense. But when the load is assisting the flexion, the neuron falls silent. So we can see that in all three conditions, the wrist displacement is the same, but the neuronal activity changes as the loads and the different muscle activities change. So the activity of the motor cortex is better related to the direction and level of forces and muscle activation are exerted rather than to the direction. So although we do have neurons that code for direction, we see that the load is the level of force, and the load is and the selection of muscles is probably creating more activity than direction and displacement. So some subsets of these neurons have unexpected or complicated responses. Another reason why it's hard to study. He's kind of showing here in Dr. Poston's text, that we see different responses we've seen at different times in the same task and in different tasks involving the same muscles. So here we see a burst of activity in a single cortical neuron during a reach and grasp movement to retrieve full food pellets. And when we study these things, we tend to have to study really simplistic movements. If we get involved with multi-joints and lots of muscles, it becomes far too complex. So a lot of neuroscientific studies are done, as I said, like on a muscle, on the FDI, something quite simple, quite a simple movement. So we see here the different muscles and their response by this movement to retrieve a food pellet. All right, so representation area, plasticity, it really is quite something. So we have these representation areas, and we can figure that out with TMS or electrical stimulation, and they rapidly change. So this is pretty interesting. As we learn things, as we learn a skill, as we play a video game, as we read Braille, as we learn to play an instrument, our motor cortex shows rapid changes with practice. And it also, by the way, shows rapid changes when we stop practicing. So when we use new muscles, we're really training our brain, and it's not something that we've thought about that way, but that's where it's originating from. And so we can see why practice is a pretty important part of motor skill learning. In fact, this is, I think that's a rat brain. Yeah, it's a rat brain. They actually find that after a body part amputation, that the brain immediately starts reorganizing itself to accommodate for the missing area. And so this is what we see in plasticity as well in humans, where people will lose part of their brain and another area of the brain takes over that function. And that's due to this, it's due to plasticity, and we can measure that in the representation area in many cases. Also, we see that there are studies in adaptation and about learning, if you will, like adaptation, learning. I suppose they're two different things, but there could be an argument that they're similar. We see that the same 4 neurons were measured over several hour experiment in a monkey, and although the kinematic neurons did not change with conditions, the dynamic ones indicated the 15 to 20 degree force field rotation. The others 2 neurons were thought to retain the motor memories. So we see that the M1 must be involved in not just the movement, but also acquisition, retention, and recall. So please do read through that. I think it's really interesting science. It will surprise and maybe, inspire you. But the take home for this slide is truly that the M1 is involved in all of these different tasks. So it's not just movement, but we also see the M1 has to be involved in learning, or we would not see this adaptation. So overall, we see there's a critical role in planning, generation, and execution of skilled movements. Our hands are extremely important. It's part of this distributed network, and it has certain areas, representation areas, and there is some overlap, and it is individualized, although we can fairly well predict the different areas. We see that it is sending an awful lot of information directly to the spinal cord, in some cases synapsing directly on the motor neurons, and that's pretty interesting. So we have direct control. Also, we see that we have populations of neurons that code for force, force direction, movement extent, movement velocity, target location, etc. It's not just this one thing. It's not just this static map of specific muscles and body parts, which you might get the impression if you look at that homunculus. So we also see that the motor cortex is also involved in adapting to novel environments. That might be considered motor learning. All right, thank you very much.